Hi, my name is Trevor Wade.
Hi, my name is Brandy Hamer. I'm in Group 7.
Hi, my name is Madison Turner, and I am in Group 7.
Hi, my name is Sebastian Despial, I'm part of Group 7.
Hello, my name is Tamara Harold, and I'm in Group 7, and we're going to be talking about cell death and how it relates to cancer.
Cancer is the over-reproduction of cells, and they divide and they cannot die. So pretty much it just causes cancer. Today we are discussing how cell death has a role in cancer. As we already know, cancer is a cause of death in many humans. Cancer is caused by cell mutations, genetics, and mutagens, but generally derives from the over-reproduction of cells. Normal cells repair DNA and the cell dies, but cancer cells pretty much just keep living and keep reproducing. Cancerous cells also ignore signals and lose molecules which break apart from each other as it is seen in this picture. So pretty much like normal cells, they are held together by molecules, but in cancerous cells, they lose those molecules, so they break apart and they spread around the body. In order for cells to overcome becoming cancerous, they must undergo cell death. Cell death helps eliminate cancerous and infectious cells to help them function properly. And the most common form of cell death is apoptosis. It is activated by factors such as clispasis, BH protein, and also BCL2 genes. Let's go into further detail. Apoptosis is cell program death, which helps cells from becoming cancerous. Apoptosis occurs when cells feel the need to die in cases of interacting with different signals and when the cell environment feels threatened. So in many cases, like when there's a lot of cells in a certain area, of course you want to kill off some of them because they're not necessarily needed. With apoptosis, it also helps to develop organisms by killing the cells, as I said earlier, to help create different parts of the organism for certain functions. So in many cases, like we've said before, like, okay, well, I may have my hand, and of course, for my fingers to function properly, I would need to kill off some of the cells so therefore they can separate. And if they can't separate, then that means that they won't be able to function as they should. In the intrinsic apoptotic pathway, the p53 tumor repressor receptor binds to the mitochondria, and in that event, BCL2 protein is inhibited and the mitochondria apoptosis complex, which is also called MAC, opens up. And when the MAC opens up, cytochrome C leaks out, and then the APAF1 protein is activated. And once that is activated, apoptosis begins.
Okay, go ahead.
In the intrinsic apoptotic pathway, death ligands such as AAFAS and transmembrane necrosis factors, they bind to the death receptor. And then the death receptor binds to the cell surface. Once the cell surface is bonded to the death receptor, it activates capaces, which are cleaved by the cytoplasm. And after the cytoplasm is cleaved by the capaces, apoptosis begins.
Hi, my name is Trevor Wade. I'm in Group 7, and I'm going to talk about BCL2 and siRNA and how siRNA is related to our topic, cell death. Okay, BCL2 is a part of the anti-apoptotic family, and it inhibits cell death. It inhibits cell death by blocking cytochrome C from entering the cytosol from the mitochondria. And if the cytochrome C goes into the cytosol from the mitochondria, it activates caspase 9 and it promotes cell death. This is basically a diagram of how the BCL2 attaches to the outer membrane of mitochondria. And the experiment we found about siRNA related to our topic was they used siRNA to silence BCL2 genes. So first they increased the sensitivity of epidermic growth factors And they did this because they wanted to speed up the process. So they bounded siRNA to BCL, and they found out that the BCL2 decreased dramatically. You see right here how BCL2 decreased from this, and siRNA is up. So by doing this, they inhibit BCL2, which promoted apoptosis, and this is basically how like the control BCL2 siRNA, the negative siRNA without the siRNA in the mock. The mock is basically the same thing as control. And the point we used was the siRNA sometimes acts as a inhibitor of the inhibitor, how siRNA inhibits BCL2 from happening. And if this does this, like I said before, it promotes apoptosis.
Hi, I'm Sebastian Desbiel, and I'm talking about pro-apoptosis. So, for apoptosis to occur, you must have pro-apoptosis signals. And there are many different types of proteins that help apoptosis progress throughout its process. The cell cannot die without pro-apoptotic signals. There are many different proteins that help apoptosis occur. These proteins are very important. Throughout this presentation, the main protein that we're going to be talking about that assists apoptosis will be the BAX gene. When survival factors decrease, death-promoting factors increase. Death-promoting proteins increase. Death-promoting proteins are also known as pro-apoptosis proteins. So, like stated right above, that's going to be the BAX, PUMA, the BAD gene. When survival factors decrease, survival factors and pro-apoptosis proteins are in a constant battle at the mitochondrial membrane. And when the death-promoting proteins outweigh the survival factors, that's when apoptosis can occur. So once the pro-apoptotic genes out-battle the anti-apoptotic genes at the mitochondrial membrane, the mitochondria then leaks cytochrome C into the cytosol, and then that progresses to caspases. And once caspases occur, that's when you get apoptosis. Now, there are other genes, however, such as BCL2, which then comes in and inhibits this right here. So apoptosis won't be able to occur. This is why it's called an anti-apoptotic gene. Okay. So the BCL2 protein inhibits the mitochondria from leaking cytochrome C into the cytosol and inhibits caspases, which means that at the end of the day, if the BCL protein does inhibit this, apoptosis will not occur. However, that's why there are BAX genes. So when BAX genes get regulated, BAX genes inhibits the inhibitor. Okay. So when the BAX gene is regulated, it ends up inhibiting the BCL2 protein, which inhibited mitochondria. So this is an inhibitor of an inhibitor, which makes apoptosis occur. Now, the BAX gene gets regulated. Okay, so the BAX gene is a tumor suppressor. Now, the problem occurs when this BAX gene does not get regulated. This can occur from post-transcriptional functions like the RNA interphase, where miRNA will then damage the BAX gene, so it won't get regulated at all. At which case, the BCL2 protein will just inhibit the mitochondria from letting cytosol C into the from letting chromosome C into the cytosol and caspases. Once this happens, apoptosis cannot occur. Okay, so now I'm going to talk about how cell signaling relates to cancer. So here are the steps that we learned, the fundamental steps of cell signaling that we learned earlier this year. The binding of a ligand by a receptor, signal transduction, and a cellular response. As you can see, all of these happen right here within apoptosis, and apoptosis is cell death, which relates to cancer. So the pro-apoptosis ligand binds to the death receptor. So that's in check. Now we need signal transduction. That occurs right here within these Cas spaces. They're relaying the signal down to let what happens in the nucleus, let it know it needs apoptosis to occur. And the cellular response is apoptosis, as you see right here. The cell is dividing up, and they're going to form different cells, and the phagocytes will then destroy it. So we have all three. So that'd be apoptosis.
Hello, my name is Tamara Harold, and I'm going to talk about P53 protein, its importance to cell death, and how it's regulated. Tumor protein P53, also known as P53 or tumor suppressor P53, is a protein that is in humans and is encoded by the TP53 gene. The P53 protein is crucial in multicellular organisms where it regulates the cell cycle and thus functions as a tumor suppressor preventing cancer. The structure of the core domain of the P53 protein is illustrated right here in light blue as it binds to the DNA, which is right here. The 6 most frequent mutated amino acids are shown right here in yellow, and these are most frequent in human cancers. All are residues of importance for P53 binding to the DNA, and this is a brief example of how it binds and how the cancer is regulated through the amino acid. In recent studies, the mutation P53 has related to cancer development. If P53 aggression occurred, it would be a crucial aspect of the cancer development as the P53 will lose its wild-type functions. Mutant P53 has also inserted a dominant negative regulatory effect on the wild type, as shown right here. Wild type control the heavy chain as it progresses in the flat P53, it decreases in the wild type. And that concludes our presentation. I hope you enjoyed our segment on cell death, apoptosis, and how it relates to cancer.